Very good. So I'm supposed to tell you about modeling, models and simulations and something like that. In the context of this class or lecture, I'm going to mean mathematical models. So how many people have actually ever done anything with a mathematical model? Just raise your hand. Something, it doesn't have to be very much. It's just I want to get a sense of whether people know what mathematical models are. So there's a lot of people who maybe haven't, right? Maybe I should get that set of hands. How many people have never worked with a mathematical model? Okay, good. So don't worry. This lecture is aiming towards you guys who've maybe never seen mathematical models, and therefore I hope you guys will follow along. And so feel free to just stop me anytime and ask questions. Okay. Since there is a mix of people, I'll sort of start very simple to begin with. But let's talk a bit about what goes into the whole process of making a mathematical model, analyzing it. So one thing already I want to emphasize is there seems to be two things you do, right? There's one thing is taking a mathematical model that for some reason has fallen into your lap. Somebody has either told you about it or you've read about it in a textbook, and analyzing it, trying to run it on a computer, trying to see maybe it's a model about, say, some ecosystem and different species, and this model tells you how the populations of different species change with time. So one thing is taking this model, running it somehow, analyzing it either computationally or analytically with pen and paper, and trying to figure out how is the population going to go. Is it going to rise all the time? Is it going to rise and saturate? Will it go up? Will it go down? Will there be oscillations? Maybe some of you have seen some of these mathematical models of ecosystems, and you can see things changing with time. So one aspect is just analyzing it, just figuring out what is going to come out of the mathematical model, and then using that to sort of look at data and back and forth. Another set of things in this whole process is where did that model come from in the first place, right? How do you build a model? What are the choices that you make in setting up the model in the first place? So I think what I'm going to do, since there are many people who haven't ever worked with mathematical models, I'm going to concentrate on that a little bit more. What are the choices? How does one make those choices in setting up a model in the first place? Actually then taking the model and analyzing it, of course, requires a lot of knowledge of various analytical and computational techniques, and I'm sure you'll learn that along the way, partly in courses, partly in classes, also from other people and reading yourself. But let's spend this 1.5 hours getting a little more sense of how does one build up a model, okay? So I sort of thought to do that, the best way would be to sort of maybe take you through an example, okay? And I'll pick an example, obviously, that I'm more familiar with. So up there on the screen, can everybody see the screen? I'm just going to run this little Java applet, which is a mathematical model of viruses and bacteria interacting. So this is a model of bacteria when they grow and they divide and so on, and they are infected by bacteriophage, okay, which are viruses that infect bacteria. And various things happen. So what you can see over here is the yellow things are the bacteria, the healthy bacteria, which are uninfected. The red ones are the infected bacteria. So what happens, an infected bacterium, a bacterium that's infected by a phage, eventually dies and releases a whole bunch of new offspring phage, okay? And these then go out and infect new bacteria and so on, and the cycle continues. And that's sort of what you see. You see an infection front sweeping through the bacteria. Once in a while, some bacterium escapes and then starts another colony somewhere else, and then there's all sorts of things going on. This is an entirely a mathematical model, okay? So it's kind of mesmerizing, which is why I chose it. But models don't necessarily have to be mesmerizing. They don't even have to be so visual. But I wanted somehow to start it off with this so you can keep in the back of your mind, maybe this is what goes into building a model? What are the processes that you put in? How do you decide how this colony of bacteria grows? How do you decide just what is happening at an infection? How does this red dot proceed and produce another red dot somewhere further? How does an infected bacterium die and then release new phage and then which go and infect something else? How do you represent all these processes? There are a number of different choices and that have gone into a model like this. And because there are a number of choices, there are also a number of assumptions. So one of the reasons I'm trying to emphasize this is in any model that you will ever make and ever build or ever analyze, there will be assumptions. And if you don't know what those assumptions are, you won't know where a model like this is applicable, where it's not, where to take its results seriously or where not to. Okay? So it's kind of important to keep in mind that there are so many choices that come about in building a model of this sort. And if nothing else, that is what I would like to put across during this 1.5 hours. You should go back feeling there are lots of choices, and never look at a model and assume that there's, it's all fine. You must know what the assumptions and choices are that have gone into it, so you know whether it applies or not in a situation you're interested in. So, okay, there's a whole bunch of stuff. We'll try to get to something like this if we can towards the end, but we need to start a little simpler, right? So what I want to do is maybe eventually head towards something like this. But let's begin by talking about some of the simplest processes happening in this kind of system. Thinking about a virus and a bacterium system, and what are all the processes we would eventually like to put in to construct something like this. Okay, so that's pretty much the end of my use of the screen, and I'll just use the blackboard from now on. Okay, so let's say we're interested in coming up with a mathematical model of how a bunch of viruses and a bunch of bacteria interact with each other and what happens to their populations over time, and what are all the different kinds of interactions that happen. Where do you start? Can we put the lights? So where do you start? वो ऑफ कर सकते हैं उसको प्रोजेक्ट. Okay, so where do we start with something like that? Let's start by, thinking about what are the questions, after all, that we want to address. Okay, it's not about just making a mathematical model for the sake of it, right? A mathematical model is a tool which you can use to answer certain kinds of questions. The questions are what are paramount. Maybe the questions you're interested in are better answered by an experiment. So let's start with the kinds of questions you would be interested in. Okay, to start with that, maybe we need to start a little bit with the biology. So how many of you are familiar with bacteriophage? Have you ever heard of them? You've all encountered them pretty much in some course or the other, right? So let's talk about virulent bacteriophage, right? You know that when they infect a bacterium, they go into this, they hijack the machinery of the cell, right? And they use it to start replicating their own DNA. Okay, and eventually they make about, does somebody know typically how many offspring phage you would expect from one infected bacterium? Do you have a clue? What kind of numbers are we talking about? So let's say some virus has infected a bacterium and it's replicating inside, and then eventually bursts the bacterium open and these offspring viruses come out. How much are we expecting? Are we expecting one virus to come out? 10, 100, 1,000? Hundreds, thousands? It's a large number, right? Let's say on the order of 100, right? I mean, I'm sure there are examples of viruses that produce a bigger burst, and there are examples of viruses that produce less, but order 100. It's a large number. Think of it from the point of view of a more macroscopic ecosystem. Think of rabbits and foxes. It's as if a fox eats a rabbit, and you get 100 new foxes. Is there something puzzling about that? What would you expect would happen if I just had, say, 100 rabbits and 1 or 2 foxes, and every time a fox ate a rabbit, say it was in this room, and every time a fox ate a rabbit, maybe a day later there were 100 foxes. What would happen? Yeah, some people are murmuring, right? You would expect all the rabbits get eaten, and then the foxes have nothing to eat. So that's a question, right? I'm just throwing out random questions here. That's a question. If you have a virulent phage and it's producing 100 new viruses each time you have an infection, why doesn't the bacterial population just get wiped out? Okay, maybe it does get wiped out. Have people done experiments like that? Well, yeah, sure, you can, right? How many of you have done experiments growing bacteria or viruses in you have. Yeah. Has anybody done an experiment where they've actually grown say in a test tube some bacteria and then put in something like T7, which is quite a virulent virus? What happens in the end? You've done something? Huh? Yeah. So the bacteria die, which is exactly what we were talking about. And yet you see in the wild, in real life, you see lots of species of virulent viruses and bacteria. In some sense, they're around. They can't have wiped out everything. So what's happening? What would you guess? Why am I spending so much time on this? We need to have some questions, like I said. You can't jump into, you're not going to do a mathematical model just for the sake of it. You are going to be addressing some questions. So we're just throwing up various questions that might come up. Okay. Very good. So now you're already hinting about evolution. Yes. So, or even there might even be, just even without evolution on a smaller time scale, bacteria multiplying. Is that enough? The bacteria multiplying, they're also being infected and the viruses are multiplying. Even without evolution of strains that are resistant, could you reach some sort of steady state? Maybe, yes, no, but that's a question. On an evolutionary time scale, is it important that new strains come up? Are they resistant? Yeah, okay, great. So various questions, right? So how do you start setting up now models to address these kind of questions? Many questions are equally well addressed experimentally as with models, but let's think about the questions that have come up here and see how you would get towards addressing them with a mathematical model. Okay, so one of the things we've been talking about, let's take the simplest question, the question that just came up. The bacteria are also multiplying. The virus is also multiplying. Could you somehow reach some kind of balance? That every time you kill a bacterium, the new bacteria are created, which fill up that loss. And there are viruses multiplying, but viruses are also dying because a virus cannot survive forever. If it's floating around and it takes a very, very long time before it finds a host to infect, it will die. It will accumulate damage for various reasons and die. So there's viruses have a lifetime, bacteria have a lifetime, bacteria have a growth rate, a doubling rate, and viruses have rates at which they infect, right? Can all this lead to some sort of stable state where both are coexisting? So now I've already, in talking about this, hinted towards some of the important processes we need to put in, right, to make a mathematical model. Talked about growth of bacteria. I've talked about infection, and I've talked about rates. So you can already see, just as soon as you start talking about these things, what we call parameters come up, right? What is the doubling time of a bacterium? What is the rate of infection of a bacterium by a virus? That's, what does it depend on? All sorts of things like that. So these are all now going to be important numbers when you build a mathematical model that you'll have to put in. You'll have to put in these numbers from somewhere. These numbers might be experimental, they might be, you might decide for some theoretical reasons that they have a certain value. Whatever it is, there are a whole bunch of numbers we need to know to actually set up this system. Okay, so let's start with the simplest part of it, or not necessarily the simplest part of it, but one part of it, just the bacteria. So have people here grown bacteria? Have you seen how colonies of bacteria grow in a test tube, for instance? Some of you have done that experiment. Yeah? Can somebody tell us how you measure how many bacteria there are in a test tube? Somebody who's done the experiment? Anyone? Yeah? Go ahead. So, okay, great. But there's something in the end you have to measure, right? So what do you measure to know how many bacteria there are? I mean, there's lots of ways, actually. I'm not saying there's only one way. Somebody said optical density. That's one way, right? Maybe there are many other ways. Maybe you can put it on a microscope and actually see them and count them by hand. That would be fine also. Okay, the serial dilution could work in that kind of situation. But fine, so there are some ways in which you can measure, right? So we could maybe, so say imagine a test tube experiment where you're growing bacteria. Do people know what the growth of bacteria looks like? Let's say this is time, and you have a certain number of bacteria you start with. This is the number of bacteria. You know how this grows? You start with, say, a small number of bacteria. How does it grow? Sorry? sigmoidal curve. So I guess it means something like that. Okay. Everyone's happy with that? Is that what you expect to happen with bacteria? Some people have seen it. Some people have actually made these curves themselves. Great. If you have, that's good. Okay. So how do you set up a mathematical model that helps you understand this curve? Okay. What happens? What are the processes you have to put in? What's going on over here? What's the, sorry? So there's an initial population size. Okay, let me just call that N0. What's the process that's happening? I mean, this is not a trick question. It's just, yeah, cell division, right? Okay, so the basic thing is cell division. Things are splitting into two, and then they're splitting further into two. How do you write that? So let's say we started at time 0 with certain number of populations, and let's say each one of these cells divide every 30 minutes. So how many cells do you expect at 30 minutes? Yeah? Everyone gets that, right? Time 1 hour. This is minutes. Yeah? And so on. Okay? People recognize this progression? Okay, good. Okay, that's a good question. In writing this, we are assuming that, right? So if you can show me a bacterium which doesn't do binary fission, it would be different. It wouldn't be this. So great, this is exactly what I want you to do. You have to, now we're starting to build a mathematical model. You have to question everything. Question whether it's a binary division, right? That's an experimental fact, for at least for some bacteria. Maybe there are situations and conditions where it's not, okay? If it is a binary division, then you're going to expect this. So some people recognize this as an exponentially increasing function, right? So what would it look like over here? It's actually going to be, so N0 and then twice and then 4 times and then 8. So it's going to go up pretty fast, something like that, right? So already we know that if you have, you can write this down as a simple mathematical model, okay? N of t is equal to twice n of t minus 30 minutes, right? This is a very, very simple mathematical model, but it is also a mathematical model. Okay, you could write it like this. This one is not enough to explain all this. Okay. Okay, that's another thing. I mean, another point over here is, if you actually looked at this and plotted this, I've sort of drawn it like this, very nice and smoothly. That's not how it's going to be, right? If it was really this process, it's going to take steps and jump. It's going to be, if this is 30 minutes, it's going to be there, like that, and then, flat, and then jump there, and so on, right? If that is how we are modeling the process. If we're saying every cell, so what's the assumption we made there? In getting this steps, in getting this thing, that everybody is dividing at exactly the same time. Now, in a typical real scenario, that's not going to happen. There's still some sense in which there is a doubling time, and the doubling time is still 30 minutes, or the growth rate is something common to all of those bacteria, but it's not doing this jump. That would happen if they started off synchronous, and if they would remain synchronous all through. So it was exactly 30 minutes for every single bacterium. Okay. Now, typically, that's not going to happen. So how does one deal with that fact? How does one deal with the fact that, for instance, it's not exactly going to be at 30 minutes? What do you do? What kind of model or mathematical process would you use to describe something where, say, the initial population, some of them are already well progressed into cell division, so they're going to divide quite quickly, and others are not so progressed, and so on. You somehow have to, you can't give them all a 30-minute, right? This is saying all the number of bacteria at t minus 30 minutes all divide and give you the new number of twice the number of bacteria at 30 minutes later. But what if some divide after 1 minute, some divide after 2 minutes, some divide after 15 minutes? You have to think of a different framework. Okay? So one of the kinds of frameworks that people use would be to say there's a probability for a cell to divide in a probability per unit time. Okay? And let's assume that's constant. You guys have heard a little bit about probability, right, in one of the earlier lectures. So suppose I say P is the probability per unit time for a cell to divide. And it's the same for all the cells in my test tube. Okay? So now, let's say at some time t, you have n bacteria. How are you going to figure out what's going to happen at some other time? 30 minutes later or 1 hour later or whatever. This is a probability per unit time. Okay? What you end up doing to deal with systems like this is you don't try to think of big jumps in time. You try to think of a really, really, really tiny, small amount of time, right? A very, very tiny interval of time, delta t, really, really small. In this time t, how many cells are going to divide? Okay? In this small time t, you can assume that each of the cells are dividing with, I'm going to write that as delta n. Now, some of you might start to see something familiar here, right? Delta n is P, how did I write? NT. Yeah? If this time gets really, really, really small, this kind of a process can be well described by a continuous process and a differential equation. I don't know how many of you are familiar with differential equations. Are you? Have some of you seen this kind of a differential equation for describing bacterial growth? Has anybody seen this kind of an equation? How more people have? A few people. Well, now you know where this comes from. And so let's recap. What are the assumptions that have gone in? We've made a number of assumptions going here, right? And you must remember this. When if you ever use an equation like that to describe bacteria growing, you have to remember what are the assumptions that went in. We assume that there is a probability per unit time for bacteria to divide, which is the same for all bacteria, right? We assumed that, we assumed you can well describe the system by the average number of cells that divide in that small time. So somehow you're taking a very, very tiny time, but nevertheless you're taking that time to be large enough that enough cells divide in that time that this is a sensible statement, that you don't have to worry about fluctuations in these numbers. This is an average quantity, right? Even in a time delta t, this is not always going to be the number of cells that divide. It's still a random process, or we're modeling it as a random process. But even if we're modeling it as a random process, this is only on average the right number. So under what conditions is this a good approximation? You need to have this number to be substantial, reasonably substantial, that you don't have to worry about the error in this number. Okay, which means typically you need to have large enough numbers or something to work with this. So what I mean is, if you have very few bacteria, if you have only like 2 or 3 bacteria, then using an equation like this might have problems. Okay? It's something you have to remember and keep in mind. Maybe you can still use it for whatever reason, but don't forget there are these kind of assumptions at the back of an equation like this. Okay? Now this kind of equation, once you have it, then you get to the second part, solving it, finding it. You can put it on a computer and figure out what it does. This is simple enough that people know how to actually analytically solve it and prove that that would give you an exponential increase. So it'll give you something like this, something continuous. Okay? So that's one sort of thing. We're starting to now build up a model, a mathematical model of bacterial growth. But compared to what you told me in the beginning about how the actual growth goes, which is this flattening out, this thing doesn't flatten out, right? So let's ask how would you put that in? Has anybody seen models of bacterial growth of this nature, which actually flatten out. Has anybody seen any of those? Has anybody heard of a logistic type equation? Yeah? I see some people nodding. So one of the ways of trying to get bacteria to grow in this sigmoidal fashion and then saturate out is by putting in something like this. Let me call it Nmax. This is something people call the carrying capacity, right? Some of you might have heard of that term. So what's happening here? Let's just for a second just look at the equation. When n is very small, this term is quite small. So you get back the old equation, right? You get back something like Pn, right? Because this equation is actually, if I open out the brackets, it's like Pn squared by Nmax, right? This is small compared to this if n is small. So we can get rid of this. So initially, when n is small, then it will grow exponentially. So you will get an exponential increase. How small is small? When I say it's small, what do I mean? Do I mean 1, 2, 10, 100, 1,000? Less than? Much less than? Much less than 1. Okay, any other guesses? How do we know? What is the right way of thinking about what is small here? It'll depend on the nutrient medium, yes. Okay, so which thing will it depend on? The nutrient medium is represented somehow in this equation, yes? Where is it represented? I mean, it's actually probably represented in, the whole environment is represented both here and here somehow. The growth rate, the nutrient will certainly affect the growth rate. Is everybody clear with that? P will effectively be something like the growth rate. Yeah, P is related to the doubling time. The higher P is, the faster this exponential, right? So basically the larger P, the quicker the doubling. Nmax is sort of telling you something about where it saturates. Okay, that's fine. We'll come back to that question of what is small. Okay, let's see the other end. Let's see near Nmax. Near Nmax, what happens? This whole term goes to 0. Yes? Everyone's agreed, right? As you approach Nmax, this goes to 0, which means this is 0. This can be thought of as the rate of change, rate of increase of the bacterial numbers. That will go to 0. So that's what's happening here, right? The rate of change is fast over here, gets slower, 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 and finally not changing. Okay? So, in effect, actually, this is where it turns out. Nmax turns out to be something like what sets that level. Okay? Okay. So now you know at the two ends what's happening. When n is small enough, and we haven't resolved that question, it's going to grow exponentially. Then as it starts getting close to Nmax, it should slow down and eventually stop growing once you're close enough, or you reach Nmax, right? So now the question is what is small enough? So the clue is over here. I actually told you that I'm going to throw away this term, right? When n is small, I'll throw this away. So that's the comparison you need to make. You need to say, is this a lot bigger than this or not, right? So n is small if this holds. So you can actually rearrange this and figure out at what value of n. So you're right, it does depend on the nutrient medium. It does depend on the carrying capacity and so on. Okay? So what sets a small or a large n in this kind of thing depends on the rates of growth and the carrying capacity and all these kind of things. Okay? So suppose you're always in a situation in your experiment where there's plenty of food, you're nowhere near this, then you're quite justified in using just this part of the equation. Okay? But you need to know where does that number lie. And where is that assumption good and where is it not? So when you make choices, this is again back to choices, right? Would you use the old equation or would you use this equation? Well, it really does depend on whether you're only interested in this part or you're interested in that part also. Okay? Okay. Where did this come from? Why this form? So it's fine. This is a form that slows down and stops. But there are so many other things I could write, right? Maybe you could write something that slows down as n gets larger, right? Maybe I could put something like this. I won't call it n max, but that's some constant. I could have something like exponential of minus alpha n. Yeah? Do all of these satisfy that property? We wanted something, remember, that starts off as being exponential. So when n is small enough, whatever small enough means, when n is small enough, these things should all reduce to this equation. Okay? Does that work for this one? When n is small enough, in this case n being small enough would mean much smaller than k. Then it doesn't matter if k is like 10,000 and n is 1. It's not really going to matter too much whether n is 1 or 2 or 3, right? So when n is small enough, you get something that looks like that. Not necessarily p. This p and this p could be different, by the way. Huh? Don't get confused. These are all different numbers. Does it work for this? When n is small enough, this is pretty close to 1, and you are somewhere near Pn. So that's fine. The other part of it is that it should go down as n increases, right? We wanted something that grows exponentially in this part, but then slows down. The growth slows down. So does it slow down as n increases? Yeah? Everyone's happy with that? Don't. I might make a mistake here, so watch out. Right? So these are other choices. Why not use these? Where does this come from? Where does something like this come from? It's not obvious. Sorry? Yeah. Well, that's okay, but what is n max? Maybe, well, yes, but then we're already assuming something about the system, that there is a maximum. Do we know that for sure? Maybe there are systems where you don't know. I mean, this is going to reach some. So let's see. So that's a good question. You're saying if you use these ones, and I used an equation like this, then it wouldn't be the correct equation because it wouldn't stop at some finite level. It would keep increasing, right? Okay. Okay, so as a function of time, you expect it to, we agree that it'll start off exponential, we agree it'll slow down. Is it going to settle down to a finite level, or is it going to sort of keep increasing and never really set? I mean, maybe go slower and slower, but it still keeps going to infinity. Huh? It'll come down. What will come down? You're saying something will come down like that? The exponential will, but that's the rate of change of the thing, right? So if this becomes 0 at some point, or very close to 0, it's still not negative. So nothing is going to go down. N is not going to go down, right? The rate of change will go to 0. Yeah. Yeah. No, you're right. The rate of change will go to 0. And I've sort of set it up so in both cases, in fact, in all three cases, the rate of change goes to 0 eventually at some point as n gets larger. But it's a valid point. How do you know that with this equation, it actually gets to some maximal value and stays there? There are lots of functions, and perhaps you know some of these, where the rate of change gets slower and slower and slower, but it never actually reaches some saturating value. It goes to infinity. Log of x is one such. The rate of change of log x gets slower and slower and slower. Okay? X to the power half is one such. It'll go to infinity. Okay? So, I'm talking about the population here. Is the population going as log of time, or is the population going as t to the power half? Not the rate of change. We know the rate of change is going to 0. But what's happening to the actual population? So that's a perfectly reasonable question. You have seen in your data, let's say you've decided from lots of experiments and data that your bacterial population reaches a finite maximum value. It doesn't keep going as you run it longer and longer. Let's say you're convinced of that. Could you use that fact to discriminate between this and this? These two possibilities in this one. Here we can be pretty sure that it actually gets to Nmax and it stops there. Can you be sure about that? Well, so that's a matter of solving the thing. Okay? But that's a perfectly reasonable way of going about it. So perhaps you'll be able to get rid of one of these. Actually, I don't think you will. I think both of these will hit some maximum and not keep increasing. Okay? But that's a matter of checking, either on the computer or analytically by pen and paper. We can figure out whether it'll keep going up or it'll hit some steady value. Let's assume it doesn't. Let's assume in all of these cases they hit steady values in all three cases. Then why do you choose this instead of that or that? Why has anybody in the literature suggested this as a good equation? Why didn't they equally well suggest that or that? Okay? So maybe they are related. Yeah, maybe there's not so much difference between them. That's one way of resolving this choice. Very possible. There's one way. You could actually argue that this one, when n is small enough, something like, let me try and rub something out here. This thing is almost equal to something like 1 minus n multiplied by some constant. You can figure out what the constant is. Again, when n is small enough. Okay? But n is small enough now in a different way. Now we're talking about you're comparing 1 over n plus k with something like 1 minus n multiplied by some constant plus some other. Has anybody heard of Taylor expansion? Never mind, don't worry about it. But there are, this can be written as something like this, where the next term is something like n squared and n cubed and so on. So you can argue somewhere that this, when n is small enough, these are small, and this is very similar. So yes, it's possible that under certain kinds of parameter ranges, certain values for k, p prime, p, n max, these two equations are really so similar that it doesn't matter. Then it really doesn't matter whether you're choosing that or that. Okay? But where's the so partly, so we've already now established something. One thing that sort of came up from that question over there is that one way of distinguishing different things is to actually go back and look at the data and see which fits better. That's certainly a good way of making some of these choices. Another way is to think about what kind of mechanisms produce these things. Okay, so where have these terms come from? Okay, right now I just pulled it out of a hat. What do you think is actually happening, and how would you represent the mechanism? So why is the growth slowing down? Resource limitation, maybe. Space limitation, maybe. All sorts of things are possible, but let's go with resource limitation. So why are we having an equation like this instead of maybe having two equations, one for the bacterial growth and one for the resource? Right? So how would one set up something with a resource? Let's try something simple-minded. There's some resource, let's say there's what's running out is carbon source or oxygen or something like that. There's something that's producing this resource, or there's a certain amount of this resource available, and it's being used up, right? It's being used up by who? The bacteria itself. So you could say that, for instance, here's a simple example. There'll be some sugar you've given to these bacteria in the beginning, and these bacteria, as they're growing, are consuming the sugar. So what kind of term would you put here? What's happening to the sugar? Now, R represents the sugar. What's happening to the sugar? It's decreasing. And what kind of term do you think I should put there? There's not necessarily a right answer to this, okay? We're building some simple scenarios. In a real situation, these are exactly the kinds of questions you'll come up with. But let's make some assumptions. Should be proportional to the population of bacteria. That seems reasonable, right? You're there's an assumption there already. You're assuming all the bacteria are consuming this sugar. All of them are sort of consuming it, say, equally. And so somehow your net rate of consumption is proportional to the number of bacteria. So even in writing something like this, there are assumptions, okay? Okay, now there's a slight problem here. Can you spot what the problem is? This is okay. This takes care of, say, this is one way of taking care of how the bacterial numbers will affect the rate. But is there any way in which the R itself will come in on the right-hand side? N is a function of R, that's good. That will come up over here, let's say. So, of course, depending on how N is changing and so on. But all that goes into N indirectly. But can R directly come in? So what if, for instance, there's no resource? And there are 100 bacteria. What's the rate at which the resource goes down? There's no resource. All the sugar is used up. There are 100 bacteria. What's the rate at which the resource will go down? Let's say R is the concentration of sugar. Let's say when R is 0, then at what rate does it go down? 0, because it can't go down. There's nothing there. So you need something that is 0 when R is 0, at least, right? There's a physical limitation there. If it's not there, it can't be decreased. Whereas here, I could, with this equation, I could give you an example where I could tell you, oh, there's none of the sugar, but there are 100 bacteria, and you'd say the concentration goes down. So that's not good enough. We need something here, something that depends on R, which goes to 0 when R goes to 0, right? You can just put R, okay, for instance. Yeah? Fine? Okay. So that's one possibility. And then up here, what will you put? There is still a growth rate. So what's happening up there? What kind of equation would you put? So now you have bacteria. They're dividing. Let's say they have a certain probability per unit time to divide. But now there is this resource around. So where does the resource, let's go back to the simple kind of equation we had. This one, right? We had this kind of equation. What's the best way? What is a way? Let me not say best. What is a way of modifying this equation to take into account a resource, R? Which bit would change? Would you add something? Would you change something? Would you multiply something? Sorry? P is going to be a function of R. Does everybody sort of agree with that? What's the logic of that? Somebody else. What does it mean for P to be a function of R? Okay, what kind of function of R? Let's say it is a function of R. Does it increase with R or decrease with R? It'll increase with R. Why? What's the logic? More resource, more growth rate. Somehow you're sort of arguing that the resource is converted into energy, and the energy changes, makes the cells divide faster and faster, right? There's a lot of assumptions there. But that's okay. That's fine. And it's probably true, right? So P can become a function of R. P is some function of R now. And it's some increasing function. Okay, let's for now just make it simple and multiply it by R. That's an increasing function. Okay, this is really not going to work actually. I can already tell you. Okay, so we have now an equation like this. Yeah. So, do you think this will give something like this? Maybe it will. Does it have the right properties? Is anything going wrong? Just by looking at it a little bit. Do you spot any obvious problem? There's not necessarily an obvious problem. I actually haven't written down this equation before, so I don't know. So there's a growth. So what can you expect will happen? You start with a certain amount of resource. You start with a certain amount of n. We really should run this on a computer or solve it analytically and check. But let's see how much headway we can go by just looking at it. So you'll be growing at some rate, and this will be going down, right? So what's going to happen to this? The resource will sort of start somewhere, and it'll sort of probably go down. Actually, it'll go down quite fast, right? If n was fixed, and it was just some constant multiplied by r, you know it would go down exponentially. Sort of a very similar equation to the old equation we saw, which had a plus sign, and things were going up exponentially. Things will go down. But you know that n is also increasing. So this constant is not constant. So it's going to go down even faster. So it's going to go down really quickly. That's exponential. It's going to sort of go down really fast. Yeah, okay, that's fine. As it goes down, as r goes down, this quantity is going to go down. PR is going to go down. So you do expect the growth rate to start stalling, right? Yes, you do. It's true, you do. So initially, the growth rate will be something, and then of the bacteria, and then it will start slowing down. So maybe this could work. You can go and check. You can try to figure out how do you actually run this on a computer, or try to solve it analytically and figure out what do you actually expect to see. Do you expect to see this? Or will it run into some of those problems we talked about where it slows down, but it keeps increasing and never actually stops? Okay, that's fine. So now you see we can start to build more and more complicated sets of equations once you start thinking a little more in detail about what's going on. So which is the better model? Let's say this model gave you a very nice fit to this data. And this model also gave you a very nice fit to that data. Why would you choose one or the other? Should you always choose this? Should you always choose that? Does that even make sense? These are questions you will have to face, okay? Already, even in making this, so if you tell me this is better than that, because it takes into account the resources and so on, and let's say we did this properly and we actually figured out just how the growth rate depends on the resource, figured all that out, and actually made a nice little equation, and it all fitted very nicely. Then I could tell you, so, and then you tell me that's obviously got more detail, and it's got more of what's going on in reality. So it's a better model. Would you agree with that statement? I can see that some people think it's a trick question, so nobody wants to commit. It's not, I mean, there are arguments both ways. I'll say that already. I often make models that are much simpler than reality. But it's not a bad argument to say that this contains more stuff, and therefore this will be better. But then I can sort of go on, right? I mean, one difficulty with that argument is I can tell you, oh, even this doesn't contain everything. You should have actually gone and figured out how is the resource converted into energy and how it's converted in. You should put that in, and you should put in every metabolite and all the whole thing, right? So what dictates choices like this? Well, what dictates is the kind of question you're asking, okay? What are you actually asking? What do you want to understand about the system? Do you want to understand exactly how the metabolic chain is made up and regulated? Is there a regulation at one step in the chain rather than at another step? Then you do need to put that in. If you're not bothered about that and you're sort of just roughly going to use this as some coarse way of understanding that bacteria grow and then they stop growing after some time when they've run out of space or they've run out of nutrients, because you're interested in something else. You remember the virus bacteria game? Maybe what we're not so interested in that thing, so interested in the precise details of what's happening inside a bacterial cell as it's growing. We're worried about what happens when there's a growing something and it's growing for some reason and it's doubling and there's some rate, and that rate might change with time. But what happens when that is attacked by a parasite or a virus? Then maybe you don't care. Or maybe you want to find out what aspects of that dynamics doesn't depend on the details of the metabolism and exactly how the food is used. In that case, maybe a simple model is good enough. Okay. But those are the kinds of choices that come up. Okay. I mean, my aim here is not to tell you what's right to do and what's not to do. I want to make you realize that there are these choices, and you are going to have to face these choices if you're going to make a model. And you should be aware of them, and you should be able to argue for whatever choice you make. Okay. In some cases, it might be sensible to take a more detailed model. In some cases, it might be sensible to take a more coarse-grained model. Okay. Any questions? Maybe I should stop a little bit here and take a drink of water and ask. Do you want to ask anything about? There were a few people, right, who have done the experiment with bacterial growth. Huh? Those guys didn't raise the question of what happens right at the beginning. You've heard of the lag phase, right? Some of you, all of you may have heard about that. When you start a new bacterial growth thing, people often talk about the lag phase, and then an exponential phase, and then it slows down. So what's the lag phase? Was there a lag phase in all this? Where's the lag phase? Sorry, yes. So, right, so here, so when you observe, you often see that it takes a while for the whole thing to start to get going. But what does that mean? Yes, it's going to take a while for it to get going already. Even if this is an exponential, it's going to start slow and it's going to speed up. So is the lag phase just a word? It doesn't really distinguish from the exponential phase? Or are you somehow saying, if you look in detail over there in time, it actually really, are you saying it's really zero for a while and then, or not zero, but let's say you started with some bacteria, really doesn't change, and then after a certain time it starts to go up exponentially? Or are you saying it starts here and starts an exponential but very slow exponential, and then at some point it switches to a faster exponential? What's going on? Okay. So, yeah. Maybe. Yeah. Okay. Very good. So, no, I just wanted to, I'm not going to get into this. But the point is, even in what we've done with the bacterial growth rate, maybe we missed some things. Huh? Maybe there are some details. Yes? Yes? Yes. Sure. And that sounds reasonable and everything. So what would you expect to happen? How would you put that into a model like this? Right? This model doesn't really take that into account. So again, these are questions you have to ask. Is that important? Is that lag phase important for whatever you're studying? If it is important, is this model sensible? Maybe it's not. Maybe it doesn't include that process. Or maybe the lag phase doesn't even exist and it's all just some exponential that's happening. Then that's fine. Then this model is okay. But you can always, so even after, 1 hour's discussion, we may still have not got into enough of the details. If you were looking at an experiment where the lag phase was important and was somehow distinguished significantly from the rest of the stuff. Okay? So you have to think about all these things. Okay. So that was a little bit about growth rate. I mean, we were planning to, get to a model of viruses interacting with bacteria. So we need to have bacteria growing. We need to have viruses sort of floating around. We need to have viruses infecting bacteria. We need to have viruses replicating inside bacteria, bacteria dying, viruses bursting. There's a lot of processes, and so far, the only thing we've talked about is bacteria growing. Okay? But that's part of the nature of things. There's a lot of sometimes interesting questions and choices, even in each of these little bits of interaction, each of these processes that you want to build into a bigger model. Okay? Yeah. Yeah. Sure. Yes. I could. Yeah. So absolutely. I mean, yeah. In some sense, I didn't get into that, but you're absolutely right. There are multiple ways. Here in the growth, I have been talking about a deterministic thing. It's some sort of average. It's something that, therefore, again, has assumptions. You're assuming that you can talk about a continuous change in the value of n, which really only makes sense if either n is a concentration and not a number, or if it's a number, it's a pretty large number, so the changes are small enough compared to the number that it's kind of like continuous. So this model, this kind of model, has that in it. In some sense, under certain assumptions, it comes from an underlying random or stochastic model. But he's absolutely right, there are situations where if you had very few bacterial numbers, or a very slow growth rate, or certain kinds of situations where this kind of equation would be completely hopeless. And so one such example would be where things were synchronous, for instance. The synchronous one, clearly this is not going to work, especially down here when the steps are sort of happening down there, and the steps are large relative to the numbers. So there are many situations where a model of this sort doesn't work. The thing I showed you, right, that growing sort of spatial simulation of bacteria growing and so on, actually doesn't use an equation like this. That just uses a rule, which has something like, and we'll come to that, we can come to spatial things if we have time. You saw that there was a little bit of a lattice like this, and there could be a bacterium occupying here and a bacterium there, and there was a red bacterium somewhere sometimes, and so on, right? And there's just a rule that sort of says, oh, if enough time has elapsed, then this one is going to double. And the new bacterium is going to come somewhere here. Okay? So that's the stochastic rule. That's the rule where you are modeling growth of bacteria, but you're actually keeping track of every bacterium. And you have you throw a dice or a random number, and you decide when it divides. Okay? So we have to get, at some point, there are again choices over there. What kind of model should you use? Should you use something like this? There are ways of extending this model to a spatial situation also. That's not the only way to do spatial stuff. So in any scenario you can make both stochastic models and deterministic models. Those are choices, again, you're going to have to make. Typically, as you probably know, whenever numbers are small, or noise, whatever is causing the noise is a substantial contribution. If noise is large or numbers are small, then it's important to get away from equations like this and look at more stochastic equations. There are many ways to make an equation like this stochastic. You could mathematically somehow add noise to this equation, okay? Or you could go to a completely different scenario and just be talking about numbers and rules for when the number increases or decreases. So, in some sense, they're similar approaches, but there are various different ways of implementing a stochastic thing. So we won't go into that in detail. But okay, so everyone gets at least the point that, again, there's a bunch of choices to make. Should you describe things in a continuous sort of deterministic sort of model, or should you describe things discretely as numbers that are changing from 1 to 2 to 4? Okay, these are choices, again, that you have to make. Okay, so let me spend the rest of the time sort of starting to add elements to this model and head towards that virus bacteria thing. And again, the focus is sort of going to be on, what are the choices that come in adding to this model. So let's stay with the deterministic thing in the first place and just see very quickly what you'd expect. Remember, we started off with what was the point. We didn't want to just do a model for its own sake. We wanted to find out, for instance, one of the questions that came up was, could you, in this process of viruses infecting bacteria and then bursting and then getting hundreds of new viruses and then so on and so forth, could you reach some sort of steady state where everybody is coexisting, whether the viruses are around and the bacteria are around, and neither is going extinct? Okay, so let's think a little about whether such a thing could exist. Okay, what do you need for something like that to exist? You need a balance between death and growth, right? So we know that every time you infect a bacterium, let's say you get 100 new viruses out. Okay? Now, think about what's the future and the fate of all these 100 viruses. If each of these 100 viruses go and infect a bacterium, they will each produce another 100, and the numbers are going to go up, as we've seen exponentially, right? It's going to be similar to the bacterial growth type of thing, except it'll have a factor of 100, so it's going to grow up really fast. So that means not all of them must be allowed to infect, right? They must die. How many must die for things to reach a sort of steady equilibrium? So one infects a bacterium, 100 come out. On average, how many of these 100 need to survive and infect another bacterium? Let's try a few numbers. Let's say none of them infect. Okay, 100 come out. For some reason, nobody infects. On average, nobody infects. So that means maybe nobody ever infects. Okay, then what will happen? Then the virus will go extinct. Okay, so that's obviously no good. That's not going to give you a steady thing. Let's try the other extreme. 100, if 100 infect, we know that's not going to be good enough. So let's try, what if 50 infect? That also seems to be a little dicey. We're going to be growing at 50, right? What if 2 infect? So you have a bacterium, 1 virus goes in, 100 come out, but only 2 of them go on to infect. And then this process continues. These, for some reason, do not find hosts and they die. All of the rest of the 100. But 2 do. 2, on average, 2 find a host. So by on average, I mean if you continue this process, then some of them will find something and this will continue. On average, there'll be 2 everywhere. What does that look like? Is the virus going to go, population going to increase or decrease or what? It's going to increase, right? It's still going to increase. It's going to increase slower than if all 100 infected, but it's still going to increase. So what do you need for it not to increase? We saw that if 0 infect, it goes down. If 2 infect, it goes up. There's not much left, right? Okay, so what happens if 1 infects? Does that work? So if really you could arrange for it, if you could somehow be in control of every single infection, you could arrange that exactly 1 infected. Then what would happen? Then it would, seems to be okay, right? Doesn't matter, they produce a whole lot of things, but only 1 infects, and so on, right? So that would lead to the numbers not increasing. So can one make that happen? Now, of course, you can't control them to be exactly 1, right? It's not, I'm not going to allow you to go in there and like be Maxwell's demon or whatever. So can you adjust parameters of the system to allow for this to happen on average? So what are the parameters? So what's going to determine this number? What's going to determine how many manage to infect and how many don't? Sorry, accessibility. Okay, what does that depend on? Let's say it's a test tube and it's well mixed. Number of bacteria present, sure. So number of bacteria should matter. Efficiency of infection. So there's some rate at which, let's say there's a rate at which you infect per, let's call this. This is the rate at which you infect, a virus infects, rate of infection per bacterium. Meaning that if in your test tube there was 1 bacterium and there was 1 virus, and you took it around, and this virus could live forever, then how much time would it take before this virus actually found that bacterium, infected it? That's sort of captured by this. Okay, and if there are more than 1 bacterium around, and we assume that everything is in a test tube, it's well mixed, so every infection is sort of independent, every bacterium is accessible, then this number would sort of tell you something about the average time you would expect between infections, right? So suppose there's 1 virus. On average, the time it would take to infect would be 1 over alpha b. Let me write that down. This is the average time to infect. Okay. So what if that these viruses, these viruses are actually, when they're outside bacteria, they're pretty robust, right? I mean, they have a protein coat, there's nothing much active going on. It's just a protein coat which contains, say, DNA or something, right? And they're pretty passive. They're just particles floating around. So suppose they actually lived forever. Suppose there's no question of them breaking up or dying or any such thing. They're just a protein coat with DNA in it. Suppose they lived forever, in the sense that it doesn't matter if they meet a bacterium 10 years later. If they meet a bacterium and they can infect it. Then what would happen? Even suppose you had very, very few bacteria, even then, as long as there's some bacteria around, you would eventually find something, right? So all of these 100 who come out, if they lived forever, they would all infect at some point. So that can't work, right? So that's the point. You need one more parameter. You need some sort of death rate of the viruses. Okay? Viruses, they have to die. These guys that are not infecting somehow have to be eliminated from the system. Whether you call that death or whether they end up going outside the system and you're only worried about some little pool and they just exit the system and nothing else is allowed to come in, or whether you actually think of it as them being damaged by chemicals or cosmic rays or whatever and actually dying in the sense that they cannot infect anymore. Somehow you have to eliminate this lot, right? So there is a time involved over there. So it's not just enough to be looking at the time to infect and the number of bacteria. You also have to be looking at the time to eliminate viruses. So let's say, I mean, again, no one needs to make assumptions here. How do they die? What happens? What is the time scale on which that happens? What are the processes that go into that? But let's assume that every virus has a certain average lifetime. Okay? So there's some sort of average lifetime of a virus. Let me call that tau. And then finally, there was one more number which you have to keep in mind, which is how many you produce, right? That was beta. Doesn't have to be 100, but let's say it's 100, around 100. Yeah. If they do not infect, correct. So if they're floating around without infecting anything, how long will they stay viable? How long will they stay in the system? Different ways of thinking about it. But there's some sort of lifetime, right? And you have that number. So now we have these three numbers, and I can't think of any other parameter that's involved. Can you? Yeah, no, it's a good point. We are now saying, in some sense, given a certain number of bacteria, we're only looking at the balance for the viruses right now. So we're looking at how many viruses you produce and how many viruses survive a burst and continue. You're totally right that one should look at this balance also for the bacteria. Okay, we'll come to that. But let's just see for a moment what's the balance for the viruses. So now, you know that, let's take one particular burst. 100 come out. Okay, you know that each of these 100 will take on average 1 over alpha b to infect. You know that their average lifetime is tau. Now one can do a very crude, very simple guess calculation for so for instance, let's assume this number is fixed, alpha is fixed, tau is fixed, beta is fixed. The only thing you have under control is how many bacteria there are. Somehow, let's say you can control how many bacteria there are and you can keep that fixed. Okay? If you could keep that fixed, what value would you choose in order to make this outcome come about? So there are 100 coming out. Okay? you want 99 of them to die. So you want basically all of them, I mean almost all of them. You want this many to die, okay? What do you need to do? Not just, yes, of course. I mean, greater than tau, but by how much? It needs to be just enough to make beta minus 1 die. Not so much. So so let's say your death rate is very, very large, okay, much larger than that, then of course more than beta minus 1 will die, okay? Come, I want you to just take 2 minutes and actually think about this. There's 2 time scales here, and there's this number, and you want that many to die before an infection happens. How many infections should happen? Only one, so that's the clue. And you know how long that will take to happen, right? You know the time, the single infection, and there's only one infection that will happen. You know how much time that will take. In that time, how many should die? That many. So now you can work it out. Yeah? So see if you get, tell me if you get this. This is what I calculated to be. 1 over tau alpha beta minus 1. Did I get that right? If you're having trouble actually doing the calculation, don't worry. Why am I doing this calculation? We have to build up a little intuition before we build up a mathematical model. So we'll have some way of knowing if our mathematical model is going in the right direction. Okay? A mathematical model should be able to recapitulate this kind of an argument. If it doesn't, and we don't get something like this, then there's something wrong with the mathematical model, right? So what are we saying? In order to have some sort of coexistence between viruses and bacteria, every time there's a burst of 100 new viruses, they can't all survive. Most of them must die. There should, on average, only one should survive in order for there to be a steady state. Okay? Otherwise, if more are surviving, the virus populations will go up. If not enough are surviving, they'll go down and hit 0. Okay? And you can achieve a balance like this if you achieved a certain bacterial density. And this is the bacterial density. If your burst was larger, does it make sense physically? You must go back and look. Does it make sense? What if the burst was 200 instead of 100? What should be the sustainable bacterial density? Should it be larger or smaller? If more bursts is happening, there are more things coming out, and you need more things to die. That means these 200 things that come out should not be able to find viruses, bacteria, hosts. So you need even less bacteria around, right? Because if you have 200 instead of 100, you're more likely to find something. So it should go down, so that's good. If the virus could infect really fast, that would tend to also mean that the bacterial population should be less, right? So increasing the rate of infection should decrease the sustainable bacterial population. And the same with the lifetime, right? If the lifetime of the virus is long, that means they can spend a long time searching. And the longer they can spend searching, the less bacteria there ought to be. So it sort of looks okay. At least there doesn't seem to be any obvious problem. Okay? So now, how can we start? Can we start getting to something like that using some model of this sort and trying to put viruses in? So let's see how you would put viruses into something like that. And again, you'll see so many choices come in. So let's say we start with something like that. Doesn't really matter which one, but I'll just start with bacteria. So bacteria are growing like this. Okay? Now there are also viruses around. So let's say there are viruses around. and those are the number of these viruses. So we'll want to have an equation for the viruses. But even before we get to that, you need to modify this equation. You need to somehow put in the fact that there are viruses, right? This doesn't take into account viruses at all. So what do viruses do? They decrease it, right? They decrease the number of bacteria. They infect bacteria. So if n is the number of healthy, uninfected bacteria, if there are viruses around, they will decrease that number. So you're going to have to put in some term that decreases it. We were talking about a rate of infection, right? Let's try to put that in. We need to put that in, because if the rate is more, then the decrease should happen faster. Yes? So somewhere this alpha should come in, right? So I'm going to propose something and you tell me if that makes sense. Sorry. I changed that. N is the same as B over here. Sorry about that. Does that make sense? So if alpha, we sort of said, was the probability or the rate at which one virus infects one bacteria. We're assuming that all the bacteria are somehow accessible and equal, so it should depend on alpha into n should be the rate at which you infect a bunch of things. But now there are many viruses. That was for one virus. The simplest case is where the virus is also doing their own stuff. Now, is there a problem? Have we made an assumption here? We have, right? We've made an assumption in some sense that the total number of infected viruses is n times, the total number of infected bacteria is n times v. What if two viruses infect the same bacteria? Right? That could happen. Or multiple viruses infect the same bacteria. In some ways, we're not taking that into account directly here, or not in any very obvious way, right? Okay, so just think about that. That's fine. Okay, but on the face of it, this doesn't seem too unreasonable. But there are, even here, there are assumptions. But let's go with this. Okay, so here's another way of thinking about it. Have people done chemical reactions and written down chemical kinetics? How many of you have done that? Two, not many. But you've done reactions like, you've seen reaction like A plus B going to C, and tried to write down how does the concentration of the product C change with time, and written equations like this. Maybe some of you are familiar with something like that. Yeah? Does this look familiar to anyone? Some people are vaguely nodding. If you are familiar with chemical reactions, think of this as a chemical reaction. A bacterium interacts with a virus, and it goes and forms some sort of infected bacteria. Okay? And we're sort of writing down some sort of chemical rate equation like thing for that. Okay? There are different ways of understanding an equation like this, and you should never be satisfied with any one way. Okay, there are many different ways of thinking about it, and all of them should make sense in some ways. If you understand what the process is, you should be able to write down something that sounds reasonable. But let's go with this. Okay, so we know how increasing the number of viruses decreases the number of bacteria. But what's happening to these bacteria? These were healthy bacteria. What's happening? So one of the things I was telling you is that they infect and then they burst, right? And they produce new things. So suppose that's what we're assuming is happening. Then the number of viruses, every time there's an infection, that should be reflected in an increase in the number of viruses, right? So, and it sort of should be the same term. But I told you that 100 new viruses come out. So maybe we need a term like this. We need beta alpha NV. Does that seem to make sense? What does something like this mean? Forget about this for now. What does something like that mean? It means in a very, very small amount of time, right? It means in a very small amount of time, delta t, these were the number of bacteria that were infected, okay? And if each of these bacteria produced beta new viruses, then the number of viruses that would increase would be beta into alpha n b delta t, right? It's just beta multiplied by delta n. So that's sort of why I'm putting something like that over there, okay? Does this make sense? Is everyone happy with this? Because you know what I'm going to do. I'm going to tell you there's an assumption here also, right? What kinds of assumptions have gone in here? Yeah. Yeah. Yes. We may not, maybe this is not necessary. You're right. In certain circumstances, it never reaches anywhere near, and maybe you're well enough ignoring this term and going for it. Perfectly reasonable question, and you can maybe once we write down the equation, that's another. It's like a homework question. You can ask, if I did put this in, and I had another model where I didn't put this in, would they show anything different? They would show something different if the viruses were not in large enough numbers, were not infecting enough that n nevertheless managed to reach somewhere close to carrying capacity. So look at something like this. If somehow there was also a carrying capacity, if this was somewhere close to the carrying capacity, if these numbers worked out to be close to that, then you would expect, maybe this is important. If it was much, much larger than that, then you, much less than that, then you would expect you could get away without it. Absolutely. Okay. What assumption has gone into this? Yes. But let's say that's something that's been measured, and it is beta is 100, or whatever number it is for a particular virus. Anything else? This is why one has to be careful while looking at mathematical models. There's lots of times where there are things which are hidden and kind of difficult. Why do I know there's an assumption there? Is because somebody told me the first time I looked at this. Okay, so what's the assumption? It's instantaneous. That as soon as an infection happens, you're bursting. That's one assumption. Actually, there are others also, but that's one big assumption. That's not how it works, really, when a virus infects a bacterium. When a virus infects a bacterium, it takes time. It needs to replicate. It needs to take on the machinery of the cell, make 100 copies. All that takes time. Often that time is on the same time scale as the growth rate of the doubling time of a cell. Okay, that doesn't necessarily mean the cell doubles while a virus has infected it. That depends on what the virus is doing to it. But it's on that same 30 minutes, 1 hour sort of time scale that you often find is the doubling time for bacteria. So here's the big assumption. You're sort of saying, I wrote this out, right? I said in a small time delta t, this would be the number of viruses infected, and this would be the number of this would be the number of bacteria infected, and this would be the number of new viruses generated. Well, maybe not. What else? Right? Go back to what we were thinking about here. There are many things we put in, but there is still something we haven't. And we only have like 2 minutes left. So let's put that in and see if we can get somewhere here. And then we can stop and we can continue discussing this. So there's something more, right? Where do you put in the average lifetime? So viruses have to die. How do you put in death of viruses? Well, it has to be a minus term, right? Number of viruses has to go down when there's a death event. So it's going to be, will it depend on the number of viruses? Let's say each virus has an independent chance of dying, some probability of dying. So if every virus has, say, 1 in 10 chance of dying in the next second, and there are 100 viruses, on average, how many will die? Always comes back to the same kind of calculation actually. That there's a certain probability per unit time, there are a certain number of viruses around. So if there were 100 viruses and each had a probability of 1 in 10 of dying in some very, very small tiny time interval, then on average, you just multiply the number of viruses with the probability, right? That's the average. So when that average makes sense to use, it's okay. So you'll get something like some number, let me call that, I don't know, alpha, beta, let's call it delta B, okay? So if there are no viruses, they can't die. There's nothing there to die. So it has to go to 0 when V is 0. And it has to be something that's proportional. Well, it doesn't have to be. I'm assuming it's going to be like that. I'm assuming they just have a certain spontaneous rate of death. Okay? So let's say you have some equations like this, right? Let's say this was a decent model of the thing. Can we recapitulate something like this? Does our intuition, does that sort of argument I made earlier still work out? How do you get that from something like this? What were we thinking about when we were thinking of this? We were thinking of some sort of steady state, right? We were thinking what will happen if the number of new viruses exactly equals the number of viruses that propagate, and therefore, and the number of viruses that die exactly make up for the number of new viruses that were created. So things have to somehow reach some steady state. Things should not be changing in time, right? So when you have something not changing in time, where is that represented here? There's no rate of change, right? So basically this is 0, this is 0, okay? And when we were thinking about that argument, we were thinking about the balance of viruses, right? So if you're thinking about the balance of virus death and birth, that's this equation, right? So let's see if this equation already gives you something like that. So you get beta alpha n v equals delta v, yeah? And if v is non-zero, then you can cancel this out and you can get n is delta by beta alpha. That starts to look somewhat a bit like this, yeah? But not exactly. So what's happened? So what about delta? We had delta here, we have tau over here. Are they related? Are they related in the right way? Does it make sense? What was delta over there? It was some sort of probability of death. And tau was some average lifetime. So it's not unreasonable that they're the inverse of each other. But they may not be exactly. There may be some number, some factor coming in or something. But okay, so that doesn't seem too unreasonable. Alpha's there, beta's there, but we had a beta minus 1. Has something gone wrong? Which is it? Does it matter? For large values of beta, it doesn't matter. Correct. Suppose now you had a virus where the burst was really only 3. Would you go with this formula or would you go with that formula? Which one is right? Can't both be right. Have we made an error in this calculation or have we made an error in setting this up? Where did a beta minus 1 come from here? Huh? 1 survives. So if you have beta coming out, you want 99 to die, not 100 to die. Okay? Okay? That sort of makes sense, right? That doesn't seem wrong. So suppose you assume that's correct, then something must be wrong here. Did we miss something? Is there some reason why this should have been beta minus 1? Now you have to start thinking of the processes that were happening, right? What was that term supposed to model? What is this equation supposed to model? It's supposed to tell you in a small amount of time, delta t, how many new viruses you get. I'll put the delta t down here. What is the net change in the number of viruses. So you have to take into account all processes that can increase the number of viruses and all processes that decrease the number of viruses. Okay? We know that if this is the number of infected bacteria and they burst immediately, that this is the net number that will come out. That seems correct. And let's forget about the death rate, that because we are worried about this. Where would a -1 come from? Is it there? Is it not there? Are you going to put, yeah? Right. Yes. So, then, anybody else? One is infecting and beta are coming out. So the net change is beta -1. So there is, I mean, so one has to be careful about these things. Okay? This is why it's kind of important to go back and forth. I mean, in a sense, that's part of the reason I was doing this calculation before making the model also. Sometimes it's good to have some expectation of what the answer is, so that when you're building the model, you know that you know when you've done it right or when you've missed some mechanism. Okay? I mean, that was a fairly crude example. I'm sure all of you would have got it eventually. And to some extent, like somebody was saying, doesn't really matter that much. When beta is large, you know, who cares? Beta -1, beta, it's all the same. But these are the kinds of sort of traps that sometimes you can fall into. You forget about some little process that you should have put in the equation. So it's important to sort of keep going back and forth and trying to do the same kind of, address the same question in multiple ways. Sometimes with a little calculation, sometimes with one kind of model, sometimes with 2, 3 different kinds of models, all of which should point to the same answer or the same question that you're studying. And it's when various things come together, then you know you're doing it right, and you know you haven't missed silly things like that. Okay? Okay, I think we're sort of running out of time. So, you know, let's just stop here.